I am eager to start the class today. So today we are going to talk a little bit more about reality rather than basic mechanisms. And there are two questions. One is how LTP and LTD may really happen in a behaving brain. And the second part, if I have time, is that I'm going to tell you that LTP and LTD are not the whole story. That if you make a system with just LTP and LTD, it will not remember anything important. Okay? So this is just to remind you of what we talked last time about LTP and LTD. Everything is, the form that we study is initiated by activation of an NMDA receptor. You know, and NMDA receptors allow calcium, and if the calcium signal is very big, you will activate some kinases and produce LTP. And if the calcium signal is not so big, it's it's the other way around. Sorry, this is big and large, and this is moderate. Okay, so if it's big and large, you will activate kinases and then leading to LTP, and if it's not so big, much moderate, you will activate phosphatase, and that's basically what explains everything that we know about the mechanism of induction of LTP and LTD. And people have demonstrated us over and over the idea of a biphasic curve with low calcium or low NMDA, LTD, high calcium, high NMDA, LTP, in many ways. One in here, for instance, is you can vary the stimulus, the frequency of stimulation, okay? And then you can see plotting here the changes. So up there are potentiations, and here depression, and here's no change. So if you do 100 hertz, as I told you at the beginning with the tetanus, you produce LTP potentiation. If you do 1 hertz for a prolonged time, you get LTD. Or you can vary the voltage of the cell. If you depolarize a little bit, but not too much, like the cells normally at -70, if you depolarize a little bit to -40, you get LTD, depression. And if you depolarize more, you will get LTP. In this case, you keep the number of pulses constant. Okay, so those are very convenient way to demonstrate that you have kind of like a biphasic curve for NMDA receptor activation, but obviously is not the way that happened in real life. Okay, so for instance, 100 hertz, the tetanus would be more akin to epilepsy than a normal event. 100 hertz for 1 or 2 seconds is actually a little bit of a bout of epilepsy. On the same token, 1 hertz for 15 seconds, it never happened like that. Unless you go to a disco, and you poof, 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 poof for 15 minutes, you get a little bit down on that. But that's not the way that you rearrange your brain. So you need a more realistic model for how plasticity happens. And I'm going to describe you so far one of the most realistic one. And here you have to keep in mind that what Hev was trying to do, it was a system in which you are accounting for correlations, basically. So this represents a pyramidal cell in the cortex, in the visual cortex, and it's taken from an experiment that I will explain probably later. So in any case, so here this pyramidal cell in the visual cortex is receiving input from the left eye and from the right eye. So normally when you're seeing things, you see with both eyes, and you see more or less the same thing. Okay? So the action potentials are kind of like very much in synchrony, not complete synchrony, but in very good register. Okay? And they drive the cell, this pyramidal cell, and also make the cells fire kind of like in the same regime. So everybody's kind of coordinated and correlated. And the idea is that neurons that fire together are wired together. So somehow this is conductive to LTP. Now, people have done the experiment that to close one eye, in this case the right eye, and you do it with a very young animal, what happens is that after a while that eye gets disconnected from the cortex. The eye is fine, but it's no longer sending information to the cortex. It's kind of like a LTD kind of thing. Okay, so it turned out that the right eye in this case is just producing very random spontaneous activity, very infrequent, and it's not in register with the other eye, which is seen in the world, and it's not in register with the pyramidal cell in the cortex because it's driven by this one. So the idea would be that neurons that fire out of sync lose their link. So you want a system, basically, or a rule that they will reward correlations, and then it punish non-correlations. Okay, and correlations is the key. Okay, people were thinking about that, but didn't pay that much attention until there was this experiment in 1995 that kind of opened the way of thinking in this way. In that experiment, you think it's unrelated, but it's very related. These people, Stewart and Sackman, what they did is, this is a pyramidal cells of the layer 5 of the cortex. Those are actually one of the longest cells in the brain, because they span the whole length of the cortex. So in a human, it can be like 2 millimeters long. So they have the soma in here, and this is the dendrite. And what they did, it was they elicit an action potential, which is this one, okay, by passing current in here. All right, that's no big deal, okay. But what they did, and that was the big deal, is that they put also electrodes in the dendrite and in the axon, okay. So they elicit the action potential in here, and the action potential traveled through the axon. That's not surprising. We know this from the time of Ramon y Cajal 100 years ago. But what was surprising is also the action potentials they were able to record it in the dendrite. So the action potential not only goes in the classical way from soma to axon, but also goes backwards to the dendrite, and it's called backpropagation. So people discovered that actually the dendrite contains sodium channels and potassium channels, just like an axon, but in less amount. Okay, so it's not capable of producing like a super beautiful action potential, but quite a decent propagating response. Okay, so if you elicit the action potential in here, it goes in here, and it also goes a little bit that way. Not all the way, but kind of like very far away. All right, and that's called the backpropagation action potential. And it turned out that that's crucial for inducing LTP. All right. So in this experiment, what they have done, following that other group, they record here, this is the soma, and they're going to elicit action potentials, and they're going to stimulate locally in here the synapses, okay, with another electrode that you don't see it in here. And what they do the synaptic stimulation, and they pair the synaptic stimulation with action potentials produced in the soma, okay? And the idea would be this action potential should propagate that way, okay? So these are the action potentials. And when they do, okay, first in an unpaired way. So unpaired or non-paired way means that you produce the action potential and then you do the synaptic stimulation separate in time, okay? And nothing happened. These are the responses, remain the same. But then they do in a pair way, as shown in here. Pair means you have the synaptic activation and then immediately followed by the eliciting of those action potentials. And in that case, you produce LTP. Okay? So if you pair, the production of action potentials in here, you potentiate the synapses that are there. And the idea would be, hey, maybe it's those backpropagating action potentials. And they show it in this figure. This is what they have done is the same experiment. Once again, this is the cell they are recording from. They put some fluorescence so you can see it better. They're going to record and evoke action potentials in here. They're going to stimulate synapses in there. But they're also going to put TTX locally in here. Okay, TTX will block the sodium channel, so it will block the propagation of the action potentials. So when they do that, forget about this make a, or this is the calcium signal that you can see a different part of the cell. Okay, 1, 2, 3, and 4. 1, 2, 3, and 4. Okay, so if you put TTX, when you put TTX in here, actually you get a lot of calcium signal in here because here is where the action potentials are being produced, but you don't get any calcium signal in here because the action potentials fail to go in there. All right. So anytime you have an action potential, you activate calcium channels and you let calcium in, okay? But now if you put TTX in here, the calcium signals are going to be here, indicating that the action potentials did not propagate all the way up, okay? Now when they do that with TTX and they do the pairing of producing action potentials in here and pair with synaptic stimulation in here, you don't get any LTP, nothing happened. You wait a little bit until the TTX goes away, all right, and then you repeat the experiment. Now there is no TTX, now the action potential can go in there, and now you get LTP, okay? So you need, in order to do LTP in here, some action potentials are produced in here and go all the way there. And that's the idea of what happened during the tetanus. During the tetanus, when you stimulate at 100 hertz, you produce a barrage of action potential that goes in there, all right? So that's kind of like the purpose of the tetanus, basically, okay? Now, it's gonna become obvious, you know, why you need those action potentials and what is the purpose of those action potentials. So in this case, this is an early experiment where they're showing this. They fill it up the cell, okay, with a calcium probe that's gonna fluoresce whenever you get the calcium in there, all right, to measure calcium, and this is the calcium signal, okay. And I think, I should see it in here. All right, so the arrow in here, it tells you where in which dendrite is. In this one, it's a bigger, and it's going to be one of those spines. Okay, so they're just going to image in a single synapse. Okay, and they're going to measure the calcium signals in a single synapse produced by synaptic activation locally in here. And in this case, this is probably a little bit of activation of NMDA receptors, okay? So you get a little bit of calcium signal. They also measure the calcium signal produced by action potentials. So at least the action potential in here is going to travel all the way up to here and branch it a little bit, and then you will see some calcium signal in here too. Very miserable, okay? But when they pair now the synaptic activation with the action potential, you get a much bigger signal, okay? And interestingly, that signal is bigger than this, and this is the summation of this one plus that one. So it's a supralinear interaction. It's just, they are not just adding, they are interacting, okay? So whatever process is evoked by synapses or by the backpropagating action potential, they are interacting to produce a big calcium signal. Okay, and that interaction is probably and obviously the NMDA receptor. Okay, and this was the kind of like the proof of that, and it's a very beautiful experiment. So what they do in here, they're measuring that the signal produced by the pairing of synapses and action potentials. But what they in very close time, like in a few milliseconds separated. But here, what they do is they invert the order. So if you activate the synapse first, and then the action potential, you get this big signal. But if you do the action potential first, and then the synapses, in a very short succession, you don't get that much of a signal. So it's very unidirectional, basically. You need the activation of the synapse first, and then the action potentials. Okay? And that makes sense if you think that what you're doing in there is to activate NMDA receptors. So the synapse is activated, you release glutamate, the NMDA grab the glutamate and is ready to be opened, but they have the magnesium block that bothered him, okay? And then the action potential came and relieved the magnesium block. And now the NMDA receptor will have like something like 30 seconds to do that because it will maintain the glutamate bound for about 50 milliseconds, okay? But if you do it the other way around, if the action potential came, the action potential is very fast. It only lasts like a few milliseconds. And if you activate the synapse, then it's not going to happen anything. Okay, you're just going to have maybe a little bit of calcium residual from the action potential, but nothing else. The NMDA is not going to fully activate. So that is what makes this asymmetry in time. So the temporal asymmetry is very defining of the need for the NMDA receptor. Okay, and now we're getting very close to Hebb, because what Hebb say it was if cell A fire an action potential and make cell B fire an action potential, then that synapse is potentiated. And that's exactly what we have seen right now. So if the synapse is activating first, then the postsynaptic cell fire action potentials, then you can get a LTP. And the converse order, if the postsynaptic cell fire the action potential before the synapse, then you should not get LTP, and most likely you're going to get LTD. All right? So interestingly here, you have the same amount of activity both in presynaptic and postsynaptic. The only thing that is changing is who fire first. Okay? And it happened. Okay? So this is a demonstration of that rule in cortex. So what we have here is a slice of cortex. They're recording from this cell that they label later with this electrode. Okay, they stimulated with this big rod. Okay, and here is a response stimulating here. They're recording there. Here's the response, and here is the time and the responses over time, 1 or 2 per minute. They varied a little bit, but they are about the same. And here, in this moment, the same response, what they do is immediately after they activate the synapse, they make the cell to fire an action potential by passing current inside the cell. Okay? So they make the cell to fire an action potential, but after the synapse was initiated, so to speak. And they repeat it for a while and then stop doing that and resume, just stimulating alone. And you can see that there was a little bit of potentiation. Now you can do it in another cell, of course, not in the same one, because that's dead. You can do the opposite, in which you record first, and then you make the action potential to precede the synaptic activation. Okay? And in that case, and then you stop doing the action potential thing, and then you produce depression. So in both cases, you get synaptic activation and action potential. It's just the order what it change. And the order, it will determine whether you get potentiation or depression. Okay? And that's kind of more neat than just blasting with a big tetanus or those things. That's more elegant. And it's hard to do just with the temporal. And here is a summary of experiment in another preparation, in which what they did is just to change the delta, the delay between the action potential and the activation of the synapse. Okay, in this case, it's first the action potential, the negative delay, and a positive delay when the synapse is first on the action potential. And you get, in this case, LTP when the things are very close, but you get nothing at about 40 milliseconds, and here also you get a lot of LTD in here and nothing at about 40 milliseconds. Now 40 milliseconds is going to be about the time, the duration of the NMDA receptor with the glutamate bound. Okay, so basically for this part at least it will be, okay, so you activate the synapse, the NMDA have a bound glutamate, and now you have 40 seconds to fire a backpropagating action potentials in order to get LTP. If the action potential is fired after 40 milliseconds, nothing is going to happen. So the NMDA receptor is detecting the temporal coincidence of presynaptic firing and postsynaptic firing. And that's exactly what Hebb was saying. So it's kind of interesting because after 50 years, the Hebb rule came back and now in full blast with the exact formulation as Donald Hebb. Donald Hebb was saying if the cell A is firing action potentials and causing the cell B firing action potentials, then you get potentiated. And it's actually, the action potentials is actually what you need to do it. Okay? So, theory sometimes goes farther than experiment. Okay? So here I'm gonna just, a summary, okay? So important is called spike timing dependent plasticity because the timing is the only thing that is code for the polarity of the changes and the magnitude. Okay, it's strictly based on the temporal correlations, not on the level of activity. And the beautiful thing is that encode for causality. Okay, so computational neuroscientists love the spike time independent plasticity because it's presynaptic cell fire and then postsynaptic cell fire is LTP, is basically indicating that pre is causing post. It's kind of like a direct reading of causality. Okay, and that is beautiful because you can inscribe causality in the world, basically causality in the nervous system, and it's powerful. So you will see hundreds, if not thousands, of papers, theoretical papers, using spike time independent plasticity as the rule for how the system learns. Okay. The other beautiful thing is, and of course the other way around. So it's the postsynaptic fire and then the pre-fire is kind of like telling that you're not causing me because I fire first without your help, so then go to hell. Okay, so it's causality and anti-causality is rewarded with LTP, non-causality is punished with LTD. Mm-hmm.
So for the postsynaptic to fire in the actual biological setting, instead of like you giving it a certain current for it to fire, how would it not depend on the presynaptic, so like the dendrites activity?
It will depend on a lot of things. So the idea is that this synapse, it will have two information basically, whether the postsynaptic cells fire and whether the presynaptic cell fire, okay, and when. And it will just do kind of like a blind, okay, this guy fire a little bit later, therefore I'm gonna do LTP and LTD. It doesn't matter, it doesn't care what it make the postsynaptic cell to fire. Maybe you need a lot of other things to happen at the same time. Maybe fire because it was just chance. Okay. So one of the, you can have some interesting consequences. How do you say that? My English is not that good. Okay. For instance, one is that it pays a lot to be part of a pattern. So if you are firing, one axon is firing and there's some other axon firing at the same time, coding for the same event, for it, so to speak, it's more likely that those guys are going to get potentiated because they're together more likely to make the postsynaptic cell to fire. Okay, that's one of the things that people, speculate about that. And the other thing is that they found also that was kind of cool, is that in order to make the system stable, because you can have a spurious coincidence. If the cells are firing all the time, it's more likely that you may have a pre and post totally by chance. So the time window actually for making LTD is a little bit bigger than the time window for making LTP. So chance, association by chance are eliminated. Only those that are really recurrent, they're gonna pop up, okay? So there's a lot of beautiful things about that people have been working and thinking. But this is basically what people are thinking now, that the plasticity doesn't happen through tetanus, but it's just by computing somehow coincidence and correlations. And that's the important variable for plasticity. So how it works? Well, I just explained you that the action potential boosts the activation of the NMDA receptor and reducing the magnesium block. How the LTD part work, nobody knows. Okay, so here I put 1, 3, and 3. 3 hypotheses that have come out that kind of explain in some cases, but not always. It's a fact, okay? So if the postsynaptic cell fire and then the presynaptic cell fire, it goes into depression, but the exact mechanism is still controversial, okay? And here there are 3. Some people say, oh, it's just the action potential came out and they leave a little bit of calcium, that with a little bit of the other calcium that of the NMDA, maybe they just add up enough to, and there are a lot of hand waving, okay? And there's other one that say, no, what happened is that the action potential desensitized the NMDA, so now it's not gonna put that much calcium, but just a little bit enough, and then a lot of hand waving, and this last one is just pure hand waving. All right, so basically, the bottom line is it happened, but we don't know. All right. So now we're gonna go to the second part. Okay, so in the first part I told you that LTP and LTD are probably happen through a spike time independent kind of rule. Now is the realization that you need other mechanisms of plasticity in addition to LTP and LTD to provide stability to the system. And people realized that, like in the 70s and in the 80s, very heavily, because LTP and LTD are basically positive feedback mechanisms and make neural networks built with LTP and LTD very unstable. So people were doing, let's make it 400 cells, interconnected with synaptic weights, let them run and see what happens if you're trying to train it. Well, the first thing that happened when you put random weights is that if the weights were a little bit on the upper side, just minimal, you tend to produce LTP, and in the next round, you're going to get more activity, and you're going to get more LTP, and in the third round, you're going to get even more activity, and then basically they start to climb up and saturate. Basically, everybody will get. It was very difficult not to do that. If you just do synaptic weights and heavy on LTP, you ended up with LTP. And if the network, for a reason, it was a little bit below that kind of like an equilibrium zero set point, it was just only 1 point, very unstable, then eventually in the next round you're going to get less activity, more likely to get LTD, and so forth, it's going to go down. So most likely it was up or down, and there was no stability. To make it stability, you had to put all of the weight into an exact value, which is impossible. So you need stabilizing mechanisms. Okay? So this is what happened. So you have LTP, you get more synaptic activity and more LTP and goes up, and then for LTD it goes the other way around. So you need something to break that positive feedback. And there are two mechanisms that can fulfill that. Okay, so you need actually a negative feedback, such as if you have a lot of neural activity, you have to reduce somehow the synaptic response or synaptic plasticity. And if you have very little neural activity, you have to enhance synaptic response. Okay, so you have to go kind of like the other way around. Okay, and one of those mechanisms came out of thinking a real experiment. Okay, so I'm going to show you the experiment. This is an experiment in the 80s. And it's about, as I told you, if you close the eye of a cat or a rat or a monkey when the animal is very young for a period of time, that eye will not be connected to the cortex. So here you have a kitten, and you activate this eye or this eye during this time, and you get, these are action potentials, very nice responses. This is my cartoon. Okay, so but if you close one eye for 2 days, all right, so what happened, and then you reopen, you evoke, you stimulate again this eye, nice, but this the formerly closed eye, you don't get that much response. Okay, and that is basically, it actually fulfills very well the spike time independent plasticity thing, because this guy is basically very randomly activated, and it's not correlated with the postsynaptic cell, and therefore get disconnected and LTD. That's perfect. Everything was working beautifully, except for this experiment. Okay, so it's a little bit more complicated. You're studying the same thing. You close one eye, okay, but then after 2 days, you do what is called reverse suture. So this eye is open, you open it and close the other one and wait. Now, the classical formulation of LTP and LTD, it will say the following. Okay, so this eye is already disconnected, so nothing is coming from there. And this eye now is closed. This guy the cortex is totally silent, basically. And nothing should happen from now on, so that you're with that poor cat. Okay. However, if you wait for 2 days, actually this eye regain full vision, so to speak. So how you can explain that, all right? And in thinking about that, there are two things that can explain that. One is the sliding threshold, and the other one is synaptic scaling. So something happening here which is not just merely LTP or LTD. So I'm going to present you the sliding threshold. Okay, that's again a very beautiful example of theory, looking before, formulating before experiment. That was done by Cooper, the guy here. Cooper was famous. I actually get the Nobel Prize for the Cooper pairs for superconductivity. Okay, so after he finished with the superconductivity, he was bored and he said, "I'm going to move to neuroscience where there's some interesting questions in there." Unlike superconductivity, which is solved according to me. Okay, so he started to think about this, and he was trying to solve this problem, and he ended up with this idea, the sliding threshold. So what is plotting here is postsynaptic activity versus a change in postsynaptic strength. So basically this is LTP and this is LTD, and there would be like a threshold. It's just basically what I have showed you so far, okay? That if you have low activity of NMDA receptor, you get LTD. If you have high, you get LTP. Actually, this guy, this theoretical formulation precedes all of those experiments. I just tell you that, okay? So that was. But he, besides predicting that this kind of biphasic curve for the plasticity, he also say that this crossover point should not be fixed, okay? And it should depend on the history of activity of the cell. So it should slide. So if the cell have been very active, producing a lot of action potentials, this crossover point should shift to the right. Or if it being less active, very lazy, then you go like here. So what happens if you have been very active, and the threshold for LTP and LTD change? Now a level of activity that before was producing powerful LTP, now it's going to produce LTD. So instead of being super active, it's going to put it down. Okay. On the other hand, if the system has been very inactive, you move the threshold for plasticity to the left. So now at a regime of activity that produces usually LTD, now it's going to produce LTP. So it produces a nice feedback. So you always come back to a set point of activity. So if activity is too high, it changes the sliding threshold. So now you can reduce the synaptic weight and go back to the set point. So it always converges to a set point. It's very automatic, and it's very beautiful in that sense. Okay, so it works. Okay, so that is one system. And so what is the idea with the kitten? So you do this, you close one eye, and then you reduce the activity, okay? And then, so you disconnect that eye, and then you close that, the other eye, and you open that one. So for a moment, that cortex is basically silent, okay? Because the only open eye is the one that is disconnected. So that caused the threshold to go all the way to a very low level. And now the minimal, miserable amount of activity that this almost disconnected eye have, now is going to be sufficient to produce LTP, and you come back. All right? Just because the threshold is sliding. Okay? So that allows you to do this adjustment and give stability. All right? So that is one case in which this theory precedes the experiment. And this is the other mechanism, which is the other way around. It's experiment, and later being formulated. And this other system is called synaptic scaling. And it's based on this observation. These round things are dishes with culture cells. So you can put neurons in culture for a while, all right? And you can record activity. So typically here it will be like this, action potentials, synaptic response, action potentials, synaptic response. And here they're both interneurons, inhibitory, and pyramidal cells. So what they did, it was they make a two type of conditioning for several days, 2 days actually. Either they put TTX or CNQX to block all activity, TTX to kill the sodium channels and this to kill the glutamatergic transmission, or they put bicuculin to kill inhibition, and therefore everything is going to be hyperactive. Okay, so there's two dramatic difference. Okay, either silence the whole thing or hyperactivate the whole thing. Then they remove the TTX and they remove the bicuculin and went back to a control thing. Okay, and what happened is in this case that the guys that were under TTX are now actually in the absence of TTX superactive, hyperactive, and this guy that were superactive and the bicuculin, now you remove it and become very low active. So it was kind of like, hey, they're trying to compensate for the lack of activity and they're pushing something to get active. And here they're so active, they're trying to push it down to get not so active. Okay? And what it changed, it was actually a glutamatergic transmission, excitatory transmission. And this is a cartoon of what had happened. So here you have a pyramidal cell with different synapses of different sizes. And what it was found, it was that after low firing rate, like the TTX or something like that, all of the synapses increase the strength. So actually they put more AMPA receptors. But all of them is killed by the same factor. Okay, so it's basically like you multiply each synapse by the same factor, let's say 2, for instance. Okay. On the other hand, if the firing rate had been very high, the easy way to control that in the dish, it was to reduce the number of AMPA receptors in each synapse, but by the same factor. Okay. And that have an interesting consequence. So the idea is that suppose that you have two synapses, synapse 1 and synapse 2, okay? And is that? Yeah. And synapse 1 is slightly bigger than synapse 2. So if you have reduced their activity and then you enhance both of them enhance, but the relation between them remain the same. And in this case, if you have increased activity, you reduce both, but the relation between them is the same. And you can show that by doing a cumulative probability of all of the events of all of those synapses, and basically if you multiply, forget about that, all right? But the whole idea is that it's a scaling factor that applies to all of the single synapses. And that's essential because remember that when you learn, the idea is that when you learn, you change the relative weight of the synapses in your neurons. So this being bigger than the other one, it may represent part of a memory, okay? Because this synapse went LTP, and later on this synapse went LTD, and you wanted to keep it like that to your memory. So now what you're doing is you're adjusting all of the weights in all of the cell by keeping the relative size the same. So basically you're keeping your memory, all right? And that's important. So you adjust the output of the cell without touching the memories. And that's crucial. That's the beauty of this system. Okay? It doesn't mess up. It changed the firing of the cell without messing up with the encoded memories in there, because the memories are encoded as a relative weight of the synapses, individual synapses. Okay? So here I am putting comparing both of them. Both of them are global. The idea is that they affect all of the synapses in the same neuron, okay? As opposed to LTP and LTD, which are totally local. LTP and LTD is computed in every single synapse, independent of the odd neighbor synapse to a great extent, okay? These two mechanisms of homeostasis, they do totally the contrary. They grab all of the information of all of the synapses in a way by computing the firing rates and make a decision and send a signal to every single synapse. In this case, it says increase or reduce the amount of AMPA receptors, and in this case, increase or reduce the threshold for LTP. I am not saying, I didn't put in here, but actually what they do is this act on AMPA receptors as a cartoon way, and this is acting on NMDA receptors. So what is going on in here is that the threshold is adjusted by changing the composition of the NMDA receptors. So if you want to increase, if you want to reduce the threshold to make it easier to do LTP, you put a more powerful NMDA receptor, basically, and you change the subunit composition to one which is more powerful, that they have more duration and pass more current, okay? Now, this other one, synaptic scaling, is acting just on AMPA. If you want to increase the postsynaptic size, you just put more AMPAs into the synapses, okay? So put right in here, so it's kind of like, this is act on the NMDA receptors, and this is writing an AMPA. And actually, there was a little bit of a spacing here, so kind of like, I think I should have foreseen that, okay? And what is beautiful is that do not affect stored memories and does not affect stored memories. So both are a control system that it will allow the adjustment without affecting memories. Okay. And I have to say that there are evidence for both of them. Okay. So what kind of experiments you could do to demonstrate whether those work or not? So actually, I participate in one of those, in this one, and my wife participated in the other one, so we both of them kind of control the whole thing. So we took advantage of the visual cortex, okay, because if you turn off the light, basically there is no more input to the visual cortex, so it's very simple to control, okay? And so you take the animal, a rat or a mouse, and put them in the dark for 2 days. Okay, so you don't have any more visual inputs. So according to the sliding threshold, you should slide the threshold towards the left to make LTP more easy, because you don't have activity. They really want to have more. Okay, so it's going to be more easy to obtain LTP, and it's going to be more difficult to obtain LTD. Okay, because you always, okay, and that's what happened. Okay. Conversely, for synaptic scaling, if you put the animals into the dark and you have less drive, you will expect it to compensate. You have to increase the postsynaptic strength. You know, put more AMPA receptors. So if you measure all of those little responses produced by a unitary activation of a synapse, they should get bigger if you put the animals into the dark. And it does happen too. Okay, so both things happen. Okay, the sliding threshold and the synaptic scaling, both of them happen. Now the big question is, aren't both the same thing? Okay, could it be that they're just kind of like the same thing? Because both of them, you know, you put them animal into the dark and it worked from both cases. Couldn't be, and here's the way that it could be. So if you put the animal into the dark and you get a bigger AMPAs, the glutamatergic response are now bigger, and then it's kind of obvious going to be much easier to get LTP. All right, because you know if the response is bigger and you're putting the same frequency now, you're going to get more likely to get LTP and more difficult to get LTP. So it will be a consequence. But the other way around also can work. Okay, because if you put the animal into the dark, now it's very easy to get LTP. Okay, so you still have some spontaneous activity, and if now it's very easy to get LTP, that spontaneous activity with that easiness of getting LTP, maybe you can produce a lot of kind of spurious potentiation, and you're going to potentiate all of the synapses. All right. So actually, it works very well both ways. So from a change in the sliding threshold, you can predict, or you can imagine that you're going to get a big responses in all of the synapses. And on the other hand, if you have the synapses bigger, it's going to be more easy to get LTP. And of course, when we realized that, I went to Leon Cooper, and I say, what do you think about that? And he say, I don't care. Because he say, it's an axiom, you know, you can start from one axiom and go to the other one. It doesn't matter. So the fact is that there is homeostasis. Okay, so the take-home message of here is that LTP and LTD are more likely devoted to detect correlations and non-correlation, and that's actually the rule for modifying the synapses. And you need these mechanisms of homeostasis to maintain the whole thing kind of stable. And there are two mechanisms, but who knows, maybe only one. All right? And you have 5 or 2 minutes if you want to make a question. No questions. Everything is clear. All right. You will see in the exam.